Hello, I am Anita Tiwari. Thank you very much for submitting excellent questions for the topic ICH-Q11 Q&A, a supporting document for the selection and justification of starting materials. I am going to answer some of these questions here. First question reads, a well-characterized small molecule drug substance was previously submitted in a drug master file and is now being submitted as part of a new DMF with no change to the manufacturing process. Will the manufacturing process be assessed against the current ICH-Q11 requirements for selecting starting material? Will there be a new starting material assessment performed? The answer is yes. In our new drug master file, it is recommended to include justification for the regulatory starting material selection per ICH-Q11 general principles described in section 5.1.1 and all the information related to the starting material, including upstream process as applicable, should be included in section 3.2.S.2.3. Next question. Is the API manufacturer required to include the route of synthesis and impurity discussion controls for the regulatory starting material in a drug master file? Yes, there may be several synthetic routes for the preparation of regulatory starting material, whether it is commercially available or custom-made. Therefore, it is recommended that API manufacturer should include route of synthesis and discussion controls of impurities, including mutagenic impurities, in the submission for a regulatory starting material procured from each supplier for better understanding of the process. Next question. Should changes in the supplier manufacturer of a starting material be reported in the drug master file? The answer is yes. Changes in the supplier manufacturer of a starting material should be reported in the DMF. Please note that all the CMC information for the starting material procured from the proposed new supplier should be included in the submission as applicable. The next question reads, why are raw materials like fumaric acid, malic acid, or citric acid not considered regulatory starting materials and subject to a higher level of scrutiny? Fumaric acid, malic acid, or citric acid are used as counter ions with the drug substance in the process. SBIA monographs are available for them. Therefore, they are not considered as regulatory starting materials. The specifications of these acids should be in line with the USP NF monographs. However, please note that the USP monograph is the minimum requirement. Since these acids are generally used in the final step to make salts, all possible impurities should be controlled in their specifications. Next question reads, does a commercially available chemical need to be manufactured under Cgmp to be acceptable as a starting material? The answer is no. Please note that Cgmp pathway starts from the first use of regulatory starting material in the drug substance manufacturing process. Therefore, commercially available chemical as starting material is not required to be performed under Cgmp. However, if API manufacturer performs purification of this material to control impurities per ICH-Q11 Q&A 5.14, they should include the purification steps of the commercially available chemical in the synthetic route in section 3.2.S.2.2 and perform under Cgmp. The specifications of pre-purified material and purified material should be included. Please note that purified material will still be considered as a starting material. Next question reads, if a late-stage intermediate is referenced in a secondary drug master file, then can we refer to this intermediate as a starting material in the primary DMF and/or application? No. Late-stage intermediate in the primary DMF should be referred as intermediate, not a starting material. Quite often we see this issue in the drug master file. You may outsource or procure this intermediate from different suppliers in the future. Also, there may not be enough Cgmp steps in the drug substance manufacturing process. Therefore, late-stage intermediate in the primary DMF should be referred as intermediate, not a starting material. Next question. What is the difference between a starting material and a key starting material or advanced starting material? There is no difference between a starting material and a key starting material. They are same. However, there is not a term as advanced starting material. It should be referred to as intermediate in the DMF submission. Thank you very much.
Hi, this is Hongliao Liao. I'm going to answer some of good questions which we received on the topic of regulatory considerations for impurity qualification. Question. Does the FDA apply ISH-Q3A for unknown impurities in peptide drug substance? Answer. Peptides are excluded by ISH-Q3A. We do not apply ISH-Q3A identification threshold for any unspecified impurity of peptide drug substance. The limit for any unspecified impurities in peptides is determined on a case-by-case basis. For certain highly purified synthetic peptide, any new impurities not observed in ROD should not exceed 0.50%. Each of the new impurities present at above 0.10% should be identified and justified. You might refer to a draft guidance ANDAs for certain highly purified synthetic peptide drug products for more details. Question. Drug substance manufacturer sets impurity limits per first drug product MDD. Second drug product manufacturer wants to use the same drug substance but has a higher MDD. Should the drug substance meet the tighter requirements, or is the drug substance allowed for the second drug product? Answer. If a DMF is intended to support multiple ANDAs with various MDDs, highest MDD should be selected for the calculation of identification and qualification thresholds per ISH-Q3A. In your case, in order to support the second drug product, the drug substance manufacturer should tighten the impurity limits to meet the tighter impurity requirements. Question. Is full validation sufficient for in-house test method for organic impurities in drug substance, or is an equivalency report also required between the in-house method and USP monograph method? Answer. If USP monograph provides method for related substance, an alternative impurity test method should be fully validated. Equivalency studies should also be performed to demonstrate that the alternative method is equivalent or better than the USP method. For the equivalency study, we need A. the multiple batches which are tested by both USP method and alternative method to show the results are comparable. B. All USP specified impurities should be included in the equivalency study unless it has been justified appropriately. For example, it's a process impurity and not possible in the manufacturing process. Question. Is it necessary to perform elemental impurity assessment again for changes like raw materials quantity optimization, batch size change, and equipment change? Answer. It's case by case. Changes like raw material quantity optimization, batch size, and equipment size changes are less likely to result in an adverse impact on elemental impurities. However, elemental impurities can be potentially introduced into the drug substance from manufacturing equipment. The risk can be reduced through process understanding, equipment selection, equipment qualification, and good manufacturing practice processes. In case of change of equipment type, for example, from glass-lined reactor to alloy reactor, you might consider repeat risk assessment of elemental impurities.
Hi, this is Dr. Yongjun Gao. I will answer some questions regarding poster 15, establishing impurity acceptance criteria as part of specifications for DMFs based on clinical relevance. The first question is, in this poster, it stated that the acceptance criterion for a specified impurity may be established at more than the SH qualification threshold if available compendial limits are greater than the SH qualification threshold. In this case, can you kindly guide us how to confirm whether higher limits are used for releasing API in marketed products? You may refer to publicly available information such as compendial monographs, scientific literature, FDA-approved package inserts, and published FDA research and assessments. The next question is, if there is no USP monograph for a drug product, the proposed limit for a specified impurity is more than SHQ3B threshold, wherein the RD limit for this impurity is also more than the SH limit. In this scenario, is it necessary to test both the RD and ANDA products using the same analytical method for the side-by-side comparative impurity analysis? In general, the same analytical method should be used for comparison of ANDA products with RD. The next question is, what's the maximum limit for total impurities in a drug substance? FDA Map 5017.2 clearly addressed this question. According to this map, the acceptance criterion for total impurities, excluding significant human metabolites, generally should not exceed the summation of acceptance criteria for individual specified impurities. These include identified and unidentified impurities. The sum total of all impurity limits, including those for significant metabolites, should not exceed thresholds that may compromise product potency and through product expiry.
My name is Donglei Yu. This question is for poster 13 on the topic of elemental impurities. The question is concerning the control of elemental impurities in drug products containing very low doses of drug substance. For a drug substance with an MDD of 50 micrograms, the limit for class 1 elemental impurities is very high. Can I use this high limit to justify the exclusion of the screening for elemental impurities in drug substance? The answer is no. We do not accept this justification. In the production of a drug product, there are broad categories of potential sources of elemental impurities, such as elements intentionally added, elements potentially present in drug substance, water, and excipients, elements introduced from manufacturing equipment, and elements leached from container closure systems. So drug substance is only one of the sources of elemental impurities in a drug product. When using the above formula to calculate the permitted concentration, please note the maximum concentration is determined using the actual drug product daily intake. You should not use the MDD of the drug substance. In your case, we recommend you use option 1. Let me reiterate this part of slide 6. We recommend option 1, using 10 grams as daily amount of a drug product. If the MDD of the drug substance is higher than 10 grams, obviously option 1 is not applicable. In this case, you may consider using MDD of the drug substance. However, please be aware that the calculated result is a maximum level for this particular elemental impurity, based on the assumption that this elemental impurity is absent from all other potential sources of the drug product. So in this situation, we recommend you talk to the ANDA applicants to set up appropriate limit. Again, ICH-Q3D is a guidance for drug product. Drug substance is only one of the sources of elemental impurities in a drug product. We suggest the DMF holder work with the ANDA applicants to determine appropriate limits for elemental impurities.
Good afternoon. My name is Barbara Scott, and I'll be covering the questions and answers on ICH-M7 hazard assessment and the main guideline. First question asks, please define what is meant by monofunctional alkyl chlorides in Note 5 of the main guideline. A monofunctional alkyl chloride is defined as a molecule containing only one alkyl chloride alerting functional group with no other alerting functional groups present. For example, on the right-hand side, we see a single monofunctional alkyl chloride. In contrast, on the left, is not a monofunctional alkyl chloride due to the presence of the nitro group. The next question asks, are other monofunctional alkyl halides, for example, monofunctional alkyl bromides, eligible for the 10-fold allowable increase in default TTC per Note 5 in the main guideline? The answer is no. The body of data that was reviewed by the ICH expert working group was specific to the carcinogenic risk that monofunctional alkyl chlorides pose to the patient, and is not applicable across other monofunctional alkyl halides. Please refer to the chapter by Brigo and Muller, reference 15 from Note 5 in the main guidelines for more information. The next question asks, you mentioned in your talk that visual inspection of a compound for structural alerts for classification purposes was not ICH M7 compliance. What does visual inspection here refer to? Could you please elaborate? This is a really good question. Oftentimes, DMF holders will submit simple statements such as, no structural alerts were identified, and there's no accompanying M7 compliant Qsar report in the submission. In these cases, the agency assumes that this statement is the result of, quote, visual inspection, end quote, which is the comparison of the impurity against literature reports that discuss structural classes associated with mutagenicity. Visual inspection can be useful as a preliminary screen for potential mutagens, but Qsar model predictions are needed for ICH M7 compliance as they provide a more accurate prediction of potential mutagenicity. Visual inspection is not M7 compliance. This question asks, can one use the AI's PDEs listed in the ICH M7 addendum for structurally similar compounds? For example, benzyl chloride has an AI PDE of 41 micrograms per day. Can one apply that limit for a structurally similar chemical like paramethyl benzyl chloride? This example is for illustrative purposes only. The answer is that the acceptable intakes in the M7 addendum apply to the impurity listed and are based on the analysis of appropriate positive carcinogenicity data as outlined in the compound-specific monograph. However, Section 7b and Note 5 of ICH M7 address the use of chemical similarity considerations in applying compound-specific acceptable intakes to other compounds. Compound-specific calculations for acceptable intakes can be applied for impurities which are chemically similar to a known carcinogen compound, provided that the rationale for chemical similarity and supporting data can be demonstrated. These justifications are consulted to the safety team in OGD and are evaluated on a case-by-case basis. If there is more than one mutagenic impurity in an API, do we need to include a combined limit for all impurities, or can an individual limit be given? We see this question all the time, and I'll refer you to Section 7 of the M7 guideline, which states that when there are 3 or more Class 2 or 3 impurities specified in the drug substance specification, a limit for each individual impurity should be listed in the drug substance specification per the acceptable intakes provided in Table 2. Additionally, in this case, a limit for total mutagenic impurities should be listed in the specification table per the acceptable intakes provided in Table 3. As stated in the guidance, compound-specific or class-related acceptable intakes and degradation products which form in the drug product are excluded from this total mutagenic impurity limit. This question asks, are cancer drugs generally exempt from ICH M7? Drug products that are intended to treat advanced cancer as defined by ICH S9 are out of scope for ICH M7. However, Appendix 1 of ICH M7 has a caveat to consider should an approved existing drug product associated with an advanced cancer indication be registered for use in a non-life-threatening indication. In this case, since the patient population and acceptable cancer risk have changed, the previously approved control strategy and limits would require re-evaluation for both the drug substance and the drug product. Question. If the impurity listed in the USP monograph is a probable genotoxic impurity based on Qsar, can we go ahead with the USP limit, or do we need to control based on TTC? Answer. USP monograph impurities that after a hazard assessment is performed are predicted positive by Qsar should be further addressed as a Class 3 impurity. An Ames test can be conducted, and if negative, the impurity is downgraded to Class 5 and the USP limit would apply. If the Ames test is positive, an in vivo gene mutation study can be performed, and if negative, the impurity downgraded to Class 5 again. Otherwise, the Class 3 impurity can be controlled by any of the 4 options outlined in the M7 guidance. We further recommend that you petition the USP for a monograph revision to have the impurity limit tightened based on a positive outcome of the AIMS or in vivo gene mutation study. The last question in my presentation asks, in cases where the intended drug product labeling may be unclear, does the agency have a mechanism for industry to request assistance for determination of the correct MDD acceptable intake prior to filing a DMF or ANDA? The answer to this question is yes. DMF holders and ANDA applicants should use the controlled correspondence communication mechanism for questions regarding MDD or acceptable intake information. Please see the link below for more information. Thanks so much for your attention.
Hi, I am David Green, and I'm going to answer a few questions we received on the following topics: control of a potential mutagenic impurity tested below 30% TTC, and Qsar negative but known carcinogenic impurities. The first question asks, if a manufacturer shows that the levels of a potential mutagenic impurity are consistently 30% below the TTC limit in 3 commercial scale batches of the API, is this sufficient evidence to omit routine control of the mutagenic impurity from the release specifications? Answer, no. Batch data alone demonstrating that a potential mutagenic impurity is consistently less than 30% TTC is not sufficient to justify no testing of that impurity. Control options 1, 2, or 3 should be utilized to test either at release or upstream in the process. Per ICH-M 7, if the option 3 control strategy, upstream control, is chosen, then 2 conditions should be met to justify this control strategy. First, a spike purge experiment at the proposed limit from laboratory scale experiments and, where necessary, supported by data from pilot scale or commercial scale batches should be provided. In addition, data from multiple batches, which is consistently less than 30% TTC, should also be provided. However, if there is negligible risk of the impurity to be present in the drug substance, an option 4 control strategy may be considered with appropriate justification. This question asks, if an impurity is found non-mutagenic by Qsar but is a known carcinogen, then what would be the classification for such an impurity? Does this carcinogenic impurity need to be controlled/evaluated at the TTC level? Answer, carcinogenic impurities that are negative in the bacterial reverse mutation assay or by Qsar analysis do not have a DNA reactive mechanism of carcinogenicity and therefore are not in the scope of the ICH-M 7 guidance. The DMF holder should refer to ICH Q3A, Appendix 3, and the accompanying notes for control of this impurity.
My name is Naomi Krulak, and I will be answering questions on the topic of ICH-M 7 quantitative structure-activity relationship, or Qsar analysis. The first question is, what software are recommended to perform an ICH-M 7 Qsar analysis? The answer is, the agency is unable to recommend specific software. However, general attributes of suitable models can be provided. Suitable models for ICH-M 7 Qsar analyses are those that are updated frequently, that would be every 1 to 3 years, where those updates include addition of new training set chemicals and mutagenicity data. A model that has an updated software interface, but a training set that predates ICH-M 7's finalization in 2014 is not suitable. Information on the age of the model and its training set can be obtained from the model's Qsar Model Reporting Format document, or QMRF. Models that are frequently updated by the developer are recommended as they generate predictions based on the most current information available. Additionally, suitable models are those that provide predictions in the form of alerting or mitigating substructural features. This provides mechanistic insight into the predictions, thereby facilitating the application of expert knowledge. Additionally, suitable models are those that use training sets that are visible to the user. This increases transparency in the predictions and facilitates the application of expert knowledge, where training set structures supporting a prediction can be reviewed by the user for relevance. In short, it is difficult to confirm or refute a prediction if it is unclear how the model arrived at that prediction, and this would be a situation where we would refer to the model as a black box. The software the agency uses for in-house Qsar analyses is highlighted in the red box on slide 8 of my March 3rd presentation. Next question. If we use a laboratory to make Qsar determinations for a DMF, does the Qsar laboratory need to be certified? The answer to that is no. The laboratory performing the Qsar analysis does not need to be certified. It is not considered a testing facility for fee-related purposes. The next question is, are Qsar model output files required in a submission? The answer to that is no, they are not required, but they are preferred. They improve review efficiency by providing unambiguous evidence of a prediction and its supporting data. In the absence of model output files, predictions may need to be rerun by the agency to confirm conclusions, thereby increasing our review time. Next question. Do impurities that are predicted to be purged from the process, for example using the tool Mirabilis, need Qsar assessment? The answer to that is yes. All actual and potential impurities most likely to arise during the synthesis, purification, and storage of the drug substance should be analyzed by Qsar regardless of whether they are expected to be purged. Next question. How often do we need to update the Qsar information in the DMF? If FDA considers a Qsar prediction shelf life of 2 years as a rule of thumb, then are DMF holders expected to reassess API impurities for ICHM7 every 2 years? The answer to that is, once a DMF application is found adequate to support a referencing ANDA, Qsar predictions are not expected to be rerun unless a specific cause for concern is identified. A specific cause for concern could be positive Ames data that becomes available for an impurity that was previously classified as negative or class 5. In such a case, only the specific impurity impacted would need to be reevaluated. Next question. A drug substance itself contains an alerting structure, an aromatic amine. It gives an Ames positive alert in Qsar, but it is known that it is not genotoxic or mutagenic. Its process impurity has the same structure, except that it has an aromatic nitro instead of the aromatic amine. How should we evaluate this nitro impurity? The answer to that is, if the impurity had contained an aromatic amine in the same chemical environment, as in the empirically negative API structure, the negative Ames data from the API could be used to dismiss the structural alert in the impurity. The impurity would then be reassigned from class 3 to class 4 through the application of expert knowledge. However, we're not talking about an aromatic amine, we're talking about an aromatic nitro group, and that's a different alert than an aromatic amine. Furthermore, an aromatic nitro compound is more likely to be mutagenic than its corresponding aromatic amine. Therefore, the negative API data cannot be used to dismiss the aromatic nitro structural alert in the impurity. However, data from other aromatic nitro compounds containing the alert in the same chemical environment may be used to confirm or refute the impurity prediction. The last question is, does the agency require hazard assessment of all reagents as well as related impurities? And the answer to that is yes, the agency requires assessment of all reagents and impurities. For reagents and impurities where Ames data are unavailable, a Qsar prediction should be used to assess their mutagenic potential. This does not include solvents, which are covered under ICH Q3C. Thank you for your attention.
Hi, this is Chanchal Gupta from the Office of Generic Drugs, and I'm going to answer some of the questions we received regarding safety evaluation of drug substance impurities in generics. The first question is, if an impurity is a metabolite of the drug substance, is it considered qualified for safety? Safety justifications for impurities which are considered metabolites of the drug substance should include both qualitative and quantitative information to qualify the impurity at the proposed level. Quantitative information such as plasma levels of the metabolite in animals or humans at the maximum daily dose that equals or exceeds the proposed clinical exposure levels should be provided to demonstrate the relevant systemic exposure to the impurity. Please refer to the Good ANDA submission guidance for more details. The second question is, can antibiotics be considered as drugs for short-term duration of use? Duration of use of a drug product should be determined based on the total number of dosing days in a patient's lifetime. Certain antibiotics may be used repeatedly over the lifetime of a patient to treat multiple disease episodes. Hence, the duration of use of an antibiotic drug product should be determined considering its clinical use over the lifetime of a patient. Please refer to the reference listed drug labeling for context of use of the drug product. I also refer you to ICH N7 guidance for additional details. The third question reads as, if a DMF supports several ANDAs, how are impurity limits in a DMF qualified for safety? Impurity limits in a DMF are qualified with respect to the referencing drug product. DMF holders should consider the context of use, that is, the maximum daily dose, duration of use, route of administration, and patient population of all referencing drug products for qualification of drug substance-related impurities. Safety qualification should include genotoxicity and general safety assessment. When setting control limits for mutagenic impurities, consider the duration of use of the product and the MDD for chronic use, that is, the highest dose of the drug administered amongst all referencing drug products. If the proposed limit exceeds the threshold of toxicological concern, a mutagenicity assessment is warranted. When setting limits for non-mutagenic impurities, the maximum possible daily dose is used to determine the qualification threshold. If the proposed limit exceeds the qualification threshold, a general safety assessment is warranted. Please refer to ICH M7, ICH Q3A, and ANDA's Impurities in Drug Substances guidances for additional details. Thank you.
Hello, my name is Deborah Johnson, and I'm going to answer a few questions on nitrosamines that we received from the webinar. The first three sets of questions were all related to the scope of the FDA nitrosamine guidance. First question, are the chemical precursors and DMFs for chemical precursors for radiopharmaceuticals, PET or SPECT, in the scope of the FDA nitrosamine guidance? Second question, should the nitrosamine evaluation be carried out for non-synthetic API? Third question, does the FDA and DMA guidance only apply to small chemical molecules but not biologics and vaccines? Answers. If the radiopharmaceuticals are chemically synthesized, then they are covered in the guidance scope. A risk assessment of the synthetic route, starting materials, and raw materials should be performed. The answer for non-synthetic APIs is no, unless there is a reason to suspect that the product contains nitrosamines, for example, between suspected contamination, a synthetic step that places the product at risk, or a vulnerable API with a functional group that puts it at risk, or process steps. For biologics and vaccines, the guidance applies to chemically synthesized drugs, which includes any drug, API, or drug product with a chemically synthesized structure, or where the manufacturing process is at risk for nitrosamine contamination due to other factors. If the biologics have chemically synthesized fragments, or the process which has a risk of nitrosamines, the guidance applies. The guidance does not include biologics that are not chemically synthesized. The next question, what level of testing and controls for nitrosamines are expected for components of container closure systems, for example, rubber stoppers? The answer, the level of control should be commensurate with the risk that nitrosamines from the packaging could migrate into the API. The FDA Guidance for Industry Container Closure Systems for Packaging, Human Drugs, and Biologics, Section 3B on Compatibility, and Table 2 of that section, shows the degree of concern with the type of drug product and the container closure components. Liquid formulations have the highest degree of concern with compatibility, as they are the most likely to be at risk for leaching and or extracting impurities from container components. Likewise, a liquid API would present a higher risk of contamination from a container closure component than a solid API. Next question. How many batches will be enough to support results for nitrosamines? The number of batches should be guided by the understanding of the risk. Nitrosamine formation in an API may be completely unrelated to the actual chemistry being used to form the API molecule. Side reactions between impurities and or reagents have often been responsible for nitrosamine formation. With a side reaction, it may be difficult to predict how much nitrosamine will form each time. Without an understanding of how much nitrosamine is being formed, it is difficult to discuss a purge strategy. Enough batches should be tested to fully justify any proposed control strategy. In general, a number greater than 3 would be expected. The next question. What is the scientific rationale behind the statement, theoretical purge factor calculations may overestimate purging factor of the process? The answer? Theoretical purge calculations are designed to be conservative. However, the purge factors, such as 1, 10, 100, or 1,000, should be based on scientific evidence. In some cases, firms assumed that NDMA and NDEA were completely miscible in water and therefore would be adequately removed from the process due to the high number of aqueous workups, and yet the API was contaminated with nitrosamines. Solubility studies discovered that while NDMA and NDA were indeed water miscible, they were also very highly soluble in organic solvents. The aqueous wash steps were much less effective than predicted at removing the nitrosamine impurities. The last question. The guidance recommends that the LOQ for all nitrosamines be less than 0.03 ppm and that routine testing be carried out when impurities are detected above the LOQ. Does the above statement mean that the decision on the need for routine testing should be based on the testing results of being less than 0.03 ppm or greater than 0.03 ppm value instead of a calculated acceptable limit based on the acceptable intake of a specific nitrosamine? The answer, the FDA nitrosamine guidance does state that the LOQ of the methods used for monitoring nitrosamines should have an LOQ of not more than 0.03 ppm. In general, the LOQ of a method should have a signal to noise of greater than 10, and it is determined experimentally. However, the agency felt it was necessary to recommend an LOQ of not more than 0.03 ppm due in part to the large number of drug products that have MDDs of greater than 100 milligrams. An LOQ of not more than 0.03 ppm was also recommended because if there are more than one nitrosamine listed in the specification, then there must be a test for total nitrosamine with an acceptable intake of 26.5 nanograms per day. In order to calculate the total nitrosamine level, the LOQ for each nitrosamine may need to be significantly lower than would be required for that limit based on the AI of the nitrosamine alone. In the case when the MDD of the drug product is very low, the nitrosamine limits may be high enough to justify an LOQ of greater than 0.03 ppm. The guidance states that routine testing should be incorporated when nitrosamines are detected at a level greater than the LOQ of 0.03 ppm. If the MDD is quite low, an argument could be presented to justify using a higher LOQ as the non-testing threshold, especially if that LOQ is between 1 to 10% of the allowable limits.
Hello. My name is Madhu Gauravaram, and I'm going to answer a few questions we received on poster number 11. The topic is review of secondary type 2 drug master files. The first question is, what is the definition of a critical intermediate? The answer is, a critical intermediate is an intermediate whose manufacturing process is deemed so important to the quality of the finished API that the manufacturing site needs to be part of the facility evaluation for the referencing application. I recommend you listen to the Drug Substance Facilities Hidden and Critical Intermediate Sites presentation given during the March 3 to 4, 2021 DMF workshop for additional information. Next question is, whose responsibility is to inform the ANDA applicant of a secondary DMF being used to source a critical intermediate? The answer is, we recommend that the primary DMF folder inform all referencing ANDA applicants of critical intermediates contained in a secondary DMF so that the facilities can be included in the applicant's 356H form. If this information is not provided to the applicant, the agency cannot convey the status of such facilities and the ANDA facility assessment may be delayed. The next question is, if an ANDA holder decides not to list a secondary DMF or critical intermediate on the 356H form, then does FDA ask the ANDA applicant to include it? If yes, when does such a query happen? Would it be asked during the RTR review cycle or during scientific review cycle? What are the factors to be considered for deciding whether a secondary DMF supporting an intermediate is needed to be listed in the ANDA 356H form? Answers for these questions are, if the critical intermediate facility of the secondary DMF is not listed in the referring application, an IR letter will be issued to the ANDA applicant to contact the primary DMF folder regarding missing critical intermediate facilities of the secondary DMF. The IR is issued after the application is found acceptable for filing when these discrepancies are discovered during the TCIR process. In some circumstances, these facilities are discovered during the full scientific review of the DMF, and a communication will be issued to the applicant in an IR or CR as appropriate. Please listen to the drug substance, facilities, hidden and critical intermediate sites presentation given during the March 3-4, 2021 DMF workshop for the factors considered in determining the criticality of intermediates, including secondary DMF sites. Thank you, and have a good day.
Hi, everyone. This is David Amsbacher. I'm a reviewer in Lifecycle API, and I gave a talk the other day on process validation. And I'd like to thank everyone for the opportunity to talk with you and all the excellent questions. We'll start off here with a question about submitting information in the drug master file. As process validation is often not completed at the time of DMF review, is it acceptable to provide a commitment to complete the process validation and submit process validation summary in response to deficiencies raised during the completeness assessment or CMC quality review? One thing to note is that the guidance states before any batch from a process is commercially distributed, a manufacturer should have gained a high degree of assurance in the performance of the manufacturing process. So that means that you must do the validation before any commercial distribution. It's possible to submit a DMF without validation data, but the guidance notes that aspects of drug substances and manufacturing processes that are critical to product quality should be determined and control strategies justified. This is the reason we ask for your validation data, because we use it to justify the limits in your process for in-process testing and impurities. And our next question asks, should both the process validation protocol and report be submitted in the DMF, or just the process validation report sufficient? The information that we use to justify the specifications in your process during our assessment is in the process validation report. So this is the most important thing to provide. All the process validation protocol information can be kept on site.
Hi everyone, I'm Xinhua Wu, one of the speakers during our DMF workshop. I'm going to answer the questions we received. The first question is, could you explain the difference between a spiked drug substance sample and a simulated drug substance sample on the slide 17, and how a suitable simulated sample is selected or designed in order to study the extraction efficiency of the genotoxic impurity from a poorly dissolved drug substance for the master validation? The main difference is, for a spiked sample, the GTIs are already in solution before being spiked to API. On the contrary, the GTIs in the simulated sample are in solid form. The slide number 17 discussed about which sample preparation strategy should be used based on the solubility of the API. for a genotoxic impurity method validation. The key for preparation of a simulated sample is to ensure sample's homogeneity. One of the strategies discussed here is to prepare a homogeneous API solution containing the GTIs at the desired levels. This can be done by adding the calculated volume of a GTI stock solution to the API based on the weight of API sample, and then dissolving the entire sample with organic solvent. The second step is to remove the solvent completely to provide the simulated sample, which is subject to analysis. To ease the preparation, volatile organic solvents are recommended. We received many questions on force degradation studies. I'm hoping the following slide can provide the answers to you. We suggest a degradation within 5 to 20% range if achievable. However, if the API is very stable, it's also understandable that the desired degradation may not be achieved. Mass imbalance within the variation range of assay is acceptable. However, mass imbalance should be explored and the explanation should be provided. Many factors may contribute to mass imbalance, such as degradants have no UV absorption or have different UV responses. Degradants are volatile. Degradants retain on the column or co-elude with API. One of questions we received is asking what can go wrong if the sample is under stress or overly stressed. An under-stressed sample may not provide adequate data for assessment, while an overly stressed sample may contain a large amount of secondary degradation products. When adopting the USP monograph methods, forced degradation study is optional. This question is asking why the upper limit of assay is used to calculate %LSD, and whether USP 621 applies to HPLC coupled with a refractive index detector. From an analytical chemistry perspective, the upper limit of assay reflects the typical analytical error, while the lower limit involves other objective factors such as water content, residual solvent content, organic and inorganic impurities, etc. Therefore, the calculation of %LSD should be based on the upper limit of assay. As to questions regarding USP 621, we suggest you to directly contact USP. For some chromatography methods that cannot meet USP 621 due to the limitation of the technology itself, justification should be provided in the DMF submission, and the proposed system suitability acceptance criteria should be reasonable based on a range of assay. This question is, does the agency recommend the exact same HPLC column used by USP when adopting the USP monograph method? If a USP method is not working for determination of related substance, how can equivalency be demonstrated? It's not agency's position to recommend if firms should use exact same USP column when adopting a USP method. Using a different column with same stationary phase is acceptable as long as the column equivalency has been demonstrated. In case the USP method is not suitable due to a different impurity profile, the firm can either demonstrate its in-house method is superior to the USP method, or develop and validate a secondary method to quantify the impurities that cannot be solved by the USP method. Questions are, we have assay and related substance only in one method. We had validated the linearity from LQ to 120% of assay. Why are we requested to validate the linearity from LQ to 120% of the spec limit using the API? How might changing the injection volume affect RF, referring to slide number 7, case 1? These two questions actually are related to the slopes of linearity curve in different concentration range. The answer for the first question is, the reference standard of the API is used to quantify individual unspecified impurities in an all-in-one method by assuming they have the same UV response. In this case, the API reference standard is deemed the external standard of the impurity as well. Therefore, validation of the linearity from LQ to 120% of the spec limit using the API is needed. In addition, the slope of the linearity curve for the same compound in the low concentration range may be different from that in high concentration range. This is usually observed when the concentration of the API sample solution is very high, especially when the analyst pushes the sample concentration to the margin in order to increase the method sensitivity for impurity determination. The case 1 on slide number 7 represents a real case how injection volume may affect the RF. The firm proposed to increase the injection volume from 20 microliter to 100 microliter, which is 5 times to the original injection volume. Increase of the injection volume will increase the concentration of analytes when the eluent passes through the cell of the UV detector. When the concentration of the analyte, in this case API, is too high, the previously validated linearity slope for the API may change, or the API peak could be saturated, and the UV response will not be proportional to the corresponding concentration angle. Therefore, the previously established RF may not be applicable. This question is, are tailing factor and the theoretical plates for system suitability of the test solution of API peak in the related substance test required? Case by case, tailing factor is recommended when the peak tailing of API is significant, or the impurity peaks are close to the API main peak, especially for peaks on the tail of API peak. As per USP 621, number of theoretical plates is only valid for isocratic HPLC methods. We refer you to USP 621. This question is asking if the firm can use its own method to determine the residual solvents based on a route of synthesis. The short answer is yes. A firm always can develop and validate its own residual solvent method based on its synthetic route. The next question is, if validation or verification of a general USP method, such as a differential scanning colorimetry test, is required. According to USP 1226, verification is not required for basic compendia test procedures that are routinely performed. However, if the test involves new or different sample handling or solution preparation, verification is recommended. In case the DSC test is used for quantitation purpose, method validation is required. This concludes my presentation today. Thank you for your attention.
Good afternoon. This is Wei Liu. I'm going to answer the questions from the presentation of common CMC issues in Type 2 DMF. The first question is, in the event that a monograph is issued following the submission of a DMF, what is the expectation of the FDA for a manufacturer to do a retrospective assessment? Answer. The basic principle is that the drug substance specification in the DMF need to be updated to comply with USP monograph requirements as appropriate. Impurities listed in the USP monograph should be controlled per USP requirements, unless justified appropriately. Impurities that are listed in the USP but cannot be formed in your manufacturing process do not need to be included in the specification, but a footnote should be added to the specification and the COA that states the impurity cannot be formed. If your original analytical methods are used for assay and impurities, and the methods are different from USP compendium method, a method equivalency study should be established between your method and the compendium method. All specified impurities in USP should be included in the method equivalency study, or justify its exclusion appropriately. In case of a dispute, the USP method will be the method of resolution. If original analytical method for assay and all impurities are changed to USP compendium method, the method verification for the compendium method should be provided. If an impurity is included in your drug substance specification, but not in the USP impurity profile, it should be demonstrated that this impurity is still controlled appropriately after the method change. The next question is, if the drug substance specification is updated during DMF or under review cycle according to the agency's review comments, could you please give an idea that how DMF holders should present the stability data summary in section S7? Answer. If the drug substance specification updates include the stability indicating tests, the drug substance stability specification in section S7 needs to be updated accordingly. When the stability study is still ongoing and the retest period for the drug substance is proposed tentatively based on current available stability data, the stability data for future time points need to be tested per updated stability specification for ongoing stability batches. When the stability study is completed and the retest period for the drug substance has been established, the data should be provided to demonstrate the drug substance at the end of retest period meet all requirements in the updated stability specification. Please be reminded all future stability data, such as annual batches, needs to be tested per updated stability specification. Question. A critical intermediate is planned to be purchased from two vendors. The vendor 1 uses the starting material A, while the vendor 2 uses the starting materials B and C to get the same critical intermediate. Is it possible to have both intermediate manufacturers with almost the same route of synthesis in one DMF? According to guidance for industry, completeness assessment for Type 2 API DMFs under GDUFA. The Type 2 API DMFs intended for reference in a generic drug submission that are subject to the DMF fee under GDUFA may only contain a single drug substance manufacturing process. The single drug substance manufacturing process means the same starting materials and the intermediates, with minor variations being allowed in solvents and raw materials, as long as the type of chemical transformation in each step is unchanged.
Thank you to our presenters for those great presentations. If you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. Looks like we have some questions coming in. The first group of questions will be directed to Dr. Krulek. And here's the first question. Why is it necessary to report the Qsar model version number?
Well, the version number indicates when the model was last updated, and it can be used to determine whether a new version of the model is now available. Most commercially available Qsar models are updated about every 1 to 3 years, and the types of updates that we generally see are expanding the model training sets to include more chemicals, adding new structure-activity relationship patterns for new chemical classes or subclasses, or improving the overall accuracy of records in the database. So we want to make sure that we're using the most current information available when we make a regulatory decision, and therefore the most current up-to-date version of the model is preferred. And also models that predate ICH-M7's finalization, so that goes back to 2014, are unacceptable. So things that are really that old are considered out of date.
Thank you for responding to that question. The next question is for Dr. Krulak. What is a Qsar endpoint? How is it defined? And how is it validated?
So this is a fairly complex, lengthy question, but I'll try and cover all parts of it. The Qsar endpoint is the specific biological effect that the model is making a prediction of based on chemical structure. A Qsar model endpoint is determined by the data that the model was actually trained on. And so an example of an endpoint could be something such as the outcome of a standardized assay, such as the Ames test, or it could be an endpoint that's a very high-level general effect, such as liver toxicity. Now, in the context of ICH-M7, the Qsar endpoint that our models need to be predictive of is bacterial mutagenicity in a 5-strain Ames test. And the model needs to be trained on data that is generated for chemicals that have actually been tested in a 5-strain Ames test. So therefore, if you had a model that was trained only on data from chemicals that were tested in a single strain, such as TA100, that is not going to be appropriate for use under ICH-M7. Now, with regards to validation, there are a number of different methods that are typically used for Qsar models. And these include recall assessments, cross-validation, and also external validation. Now, recall and cross-validation are where we use methods to see how well the model predicts the chemicals that it's actually trained on. However, external validation is when we take a completely new set of chemicals that the model was not trained on and challenge the model to predict those. And we calculate how well the model performs in each of those situations. This information about the Qsar endpoint and also the validation statistics for the model are typically going to be reported in the model's QMRF, or Qsar Model Reporting Format document.
Thank you for responding to that question. And one last question for Dr. Krulak. A chemical substance is predicted positive by the expert rule-based model and negative by the statistical-based model. The software gives a suggested overall prediction of negative. Can this overall negative prediction be accepted without further review?
So the answer to this question is no. We can't just accept that negative overall suggested prediction without doing further review. This is a case where if you look at the individual predictions, we have a positive and a negative, and that's going to default to a positive overall prediction, a class 3 assignment in the absence of any kind of expert review. But some software have this added layer to guide the user through an expert review process, and it will do an automated database search for perhaps other chemicals that contain the same structural alert or chemicals that are globally similar, and it will algorithmically use that information to generate an overall prediction. But that's being done by the computer, and that needs to be vetted by a human expert. And so for that reason, you can't just accept that suggested overall prediction without confirming that it's based on good information.
Thank you for responding to that question. The next set of questions is directed to Dr. Scott, and here's the question. Do we need to include Qsar study data for impurities in the DMF, or is it just the prediction of each model enough in a table?
Hi, thanks, Ray. That's a really good question. For the hazard assessment table that I showed in my presentation in March, I showed only the final individual model predictions, and that's really what's necessary to support the impurity classification. But the agency does prefer that the model output files also be appended to address any questions that might arise about the predictions. I also want to remind you that your Qsar supporting data gets reviewed by Naomi's group in the computational toxicology farm tox group for concurrence. And I'll just remind that slides number 18 and 19 of Dr. Krulak's talk in March outlined what the supporting Qsar report should contain. And I'll just briefly list those for you here. Materials and methods, including, as we just talked about, the software names and version, the individual model predictions and the overall classification, put that in tabular format for ease of our review. A detailed explanation of any expert knowledge that your group applied, particularly if you overturned predictions. And ideally, you should include an appendix containing the model output files and pivotal experimental data to support your overall classification. Thanks.
Thank you for responding to that question. The next and final question for Dr. Scott. What are the control strategies to be adopted for inorganic impurities which are positive for mutagenicity?
Thanks. Another really good question. DMF holders are responsible for performing a hazard assessment to classify reagents, starting materials, intermediates, potential byproducts in the process, and degradants. ICH M7 does not distinguish between organic impurities and inorganic impurities with respect to control strategies if mutagenicity is determined. Thank you.
Thank you for responding to that question. The next set of questions will be directed to Dr. Lau. And here's the first question. Is it acceptable to control only the highest impurity and not the single unspecified impurity in drug substance specification? For example, to have an acceptance criterion as the highest unspecified impurities not more than 0.2%.
The answer is no. The proposed acceptance criterion is not acceptable. Drug substance specification should include each specified identified impurity, each specified unidentified impurity, any unspecified impurity, and total impurities. For drug substance within the scope of ICH Q3A, any specified impurity with a proposed limit above qualification threshold should be suitably qualified. Any specified impurity with a proposed limit above identification threshold should be identified. When identification of an impurity is not feasible, a summary of the lab studies demonstrating the unsuccessful efforts should be included in the application. Thank you. Thank you.
Thank you for responding to that question. The next question for Dr. Lau. ICH Q3D guideline and USP have elemental impurity permitted daily exposure, or PDE, limits for common routes of administration. What PDE criteria should be used for other routes of administration, such as topical or ocular products?
That's a good question. PDEs of ICH Q3D have been established for oral, parenteral, and inhalation routes of administration. PDEs for other routes of administration can be derived according to the principles described in ISHQ3D. To facilitate the review process, we recommend that you work closely with your drug product applicants for the specific PDEs of elemental impurities. Alternatively, you might communicate directly with FDA by submitting a controlled correspondence.
Thank you for responding to that question. And one last question for Dr. Lau. And here's the last question. What types of toxicological studies are required to qualify an impurity exceeding ICH-Q3A qualification threshold?
That's a good question. And you might refer to Attachment 3 of ICH-Q3A for the toxicological studies required to qualify an impurity above qualification threshold. The studies should consider patient population and duration of use. They include genotoxicity studies, general toxicity studies, and other specific toxicity endpoints. Thank you.
Thank you for responding to that question. The next group of questions, the time we have remaining, will go to Dr. Johnson, and here's the first question. If more than one nitrosamine impurity is specified in the specification, do the individual impurities have to be not more than 0.03 ppm each?
Thank you for that question. So the nitrosamine controls, I realize, in the guidance are just a little confusing. So let me re-explain. So if you have a single nitrosamine, you are allowed a limit up to the acceptable intake based on MDD. If you have more than one, let's say you have two, you may have a limit for each nitrosamine up to the limit based on their acceptable intakes and the MDD. However, it's when you have more than one in there, we have to worry about total nitrosamine. That total nitrosamine limit is the 26.5 nanograms per day. So that is the one that has to be controlled at that limit based on the MDD. So you may have each individual up to the acceptable intake based on MDD, but if you have more than one, you also have the requirement that the total nitrosamine limit is limited to 26.5 nanograms per day based on MDD.
Thank you for responding to that question. The next question for Dr. Johnson. Is a risk evaluation of the manufacturing process required for nitrosamine's presence even after the manufacturing process has been modified to eliminate the nitrosamine formation?
Okay, great. That's a good question. So if the process has been modified to eliminate the nitrosamines, then I would assume a risk assessment had been done prior to that in order to identify what needed to be changed. So the risk assessment, is that something that needs to be kept on site? That's not something that needs to be sent to the FDA. And it's really up to the company to decide what that risk assessment entails. So in this particular instance, it sounds like there must have been some kind of assessment done because a risk was identified and then the process was actually modified. And I'm assuming that information is being kept somewhere on site. What you do need to communicate to the agency is if you've had to change the process, so there's an actual process change in S2.2, or if there's any control strategies or methods that have changed. That's what would be needed to be reported to FDA. Thank you.
Thank you for responding to that question. Well, it looks like we're just about out of time, and we'd like to thank our presenters and panelists for responding to numerous questions that came in. Now we'd like to welcome Dr. Skoda to provide us with some closing comments.
Hi, I'm Erin Skoda, an Acting Branch Chief in the Division of Lifecycle API. Thank you so much for being here today. I'd like to say a few words to close out our SBIA, DMF, and drug substance follow-on webinar. Today we covered your questions as a follow-up to our March 3rd and 4th SBIA, DMF, and drug substance workshop. The questions came from both the live Q&A session at the workshop as well as the questions that you submitted via email. We received so many thoughtful questions on all the topics we discussed, and we truly appreciate the effort you put into submitting these questions so that we could tailor our focus today to what interested you the most. The topics we covered today range from administrative aspects of drug master files to detailed information about specific quality aspects of your drug master file. We had more useful panel discussions, and the recordings from today will be available later. When we first started discussing the DMF and drug substances workshop and webinar, we had envisioned a lot of face-to-face interactions between FDA staff and the workshop attendees. While we had to change our plans for how we interacted, we did manage to have 2 events covering 3 days that achieved our goal of having as much interaction as we could between everyone, given the pandemic. This webinar is the culmination of this 3-day event, and it is fitting that it was solely comprised of questions asked by the audience. This webinar, as well as the DMF and drug substance workshop, was the effort of many individuals. First, I'd like to thank the SBIA team, Captain Brenda Stoddart, Captain Ray Ford, and Lisa Miskevich. Their guidance and expertise were very much appreciated over the last several months, and we couldn't have put on these 2 events without them. Thank you to the Division of Lifecycle API Webinar Planning Committee. Specifically, Barbara Scott, who is the chair of today's session, who is really responsible for organizing all of us and our efforts. Commander Dave Skanke, our division director, Ram Rondad, Wei Liu, Fardak Abelverdi, Qian Shi, and Yingzi Wang. Thank you also to all of the speakers, poster presenters, and question answerers. We had so many contributions from the Division of Lifecycle API, as well as our colleagues and collaborators from across the agency. And they really put forth a lot of time and effort to make sure that the content that we delivered to you would be useful and clear. Finally, I'd like to thank all of you for attending today's webinar. The registration and attendance for this workshop and webinar were record high, and we're so pleased that we were able to reach so many of our colleagues from across the world. As we close the final event of our SBIA, DMF, and drug substance workshop and follow-on webinar, we're leaving you with many hours of recorded content. All of the workshop's full talks, poster presentations, poster Q&As, follow-up Q&As are available at the SBIA website linked here. In addition to the recordings, there are downloadable slides. Please check out anything you might have missed. Thank you for being here. We really appreciate everyone who contributed to and attended today's event. Stay safe, everyone.